Hi, everybody. This is Dr. Ed Park. I'd like to talk a little bit about what I learned at the International Society for Extracellular Vesicles meeting. I hope it interests you. The recurring themes of the 5-day conference were that people were up there confessing, oh, I was wrong about this. The field is relatively young. Only about 13 years, I think the ISEV has been in existence. There were a lot of companies, over 30 companies displaying. There were 1,000 either candidates or PhDs, principal investigators from around the world, over 52 countries. And the common themes were, we wish we had a liquid biopsy to diagnose disease. Nobody really knows how exosomes are made, processed, but we're getting there. And I did learn that they're a fun crowd. It's a young field, and some of the leaders uh, have young children, like Rosella Crescitelli out of Sweden, I think. Her daughter created this wonderful Eevee, which is a Pokemon, and Eevee is also, of course, the abbreviation. And then we got to party pretty hard with the Bavarian Beer House, and then they rented out a posh disco, and these PhDs know how to party. The first day was the education day organized by these three wonderful folks, and it was a good review for people like myself. And one slide that came up quite frequently is from the MySev guidelines, and it just goes back into what is and what is not an EV. So let's review this for a second. Everything that is not a cell is an extracellular particle. They can either be vesicular or non-vesicular. If they're non-vesicular, then they're non-vesicular extracellular particles on the right. Makes sense. If they are vesicular, they can be three kinds. They can be either EVs, extracellular vesicles, synthetic vesicles, which we create, or artificial cell-derived vesicles, which is a hybrid, I suppose. Now, when we go into heterogeneity, it's important to look at this slide. This is a slide in real time. It's moving in the video. You could see from blood, there's red blood cells, and then there's platelets, but then there's a bunch of extracellular vesicles, and they come from both lymphocytes, regular cells, and from the platelets themselves, and they outnumber the cells 10 times. Interestingly, unlike with a regular stem cell culture, if you have serum, you also have this confusion with chylomicrons, VLDL, LDL, which are on the same size. Also on the same size are apoptotic bodies. They can be small, retroviruses. So there's a whole mix of things that could be in your mix, unless you can siphon or filter them out. When we look at imaging, above me, we see again that the size of an exosome around 100 nanometers is about the size of a chylomicron, which is cholesterol-filled structure. There's also VLDL, very low density, and LDL. So these things confuse the particles, and a lot of people get confused over particle counts, which are not exosomes. So how do we distinguish? Well, if you look at the density above, the LDL or liponanoparticle, or I'm sorry, lipoproteins, I should say, hard to talk, are denser, but it's not that easy to separate them. And when you're counting, you're really just counting particles, which has implications. If we look at the nanoscale heterogeneity, what that means is that size matters. You know, I guess it's like 4.3 pi r cubed, the amount of area or volume inside a vesicle. And this just does a thought experiment. If the diameter is 50, 100, 150, the relative volume increases proportionally. And the number of proteins it can handle increases. And it's important to note that even though they do single EV analysis, you really want the population, because over, a stable cell culture, if you have sampling in the billions and you dissolve them and do the omics, then there becomes predictable patterns. But keep in mind, not every particle has everything. In other words, as it says here, each particle holds a small set of the total molecules. One interesting thing that comes up is, what are the contaminants? People are very conflicted over this. If you look at the meatballs above, you got the meatballs and you got some cheese and sauce and scallions or whatever that is. And a lot of times you have co-isolates or we're not really sure what the effect of the corona, so-called corona, or the protein coding is. Sometimes it's just albumin. Sometimes people hypothesize they could have a functional role, but nobody is too sure about that. One admonition that Rosella said was that if you use ultra high G centrifugation, which is kind of the industry standard for years in published studies, they can actually form combination structures. I mean, think about if you have two bubbles and you're forming it at 100,000 Gs, I guess they could combine, and we'll see that later on. That is indeed the case. But more concerning, they can actually lacquer on the proteins from the corona to become part of that mess and form an aggregate. I also didn't know and learned that Triton X, which I talked about in the book and we talk about, although an industry standard, it can be deprecated. Some companies don't use it anymore because it's too dissolving. So whenever you have a soap to dissolve the vesicles and do your omics of what's inside the endosomes or the exosomes, you need to be careful because soaps can also damage and denature what's inside. So it's always a balance. Some people actually do not use Triton X100, I learned. Again, this slide above me was a popular slide showing all the different kinds of vesicles that can butt off for whatever reasons. Not even shown here are the tinier ones, less than 40 nanometers or so, and those are the exomeres, which they're just starting to discover. There's a strong bias between small EVs, which are formed through this endosomal pathway. These MVBs are like you see above me, batches of exosomes are excreted, and that's a nice biosynthetic pathway that creates a certain bias. But I asked a question towards the end about microvesicles, which are traditionally larger because they can't fit that many into a multivesicular body. And one of the scientists said, oh yeah, we have analyzed those, and often the contents are very similar. So even though there's a bias for the small 100 nanometer size, the 400 might just be like, coughing instead of breathing out exosomes, if you will. One of the founders of this relatively young field, I made an error in the book that I wrote. I said that it was a sleepy backwater of neurobiology, actually also even longer and more into the field decades earlier were the immunologists. So immunology actually incorporated the idea of small extracellular vesicles, and that's been decades in research. And one of the principal investigators from the Marie Curie Institute, Clotilde Thierry, was a leader of the ISEV, and there have been several female leaders, and I would say it's a young field, and I would say most of the lecturers were female, so it's kind of a female-dominated field. She gave a beautiful lecture, which was kind of above my head, saying that the canonical markers that we see, CD63 and CD9, if you want to know whether they came from the endosomal pathway or they butted off from the surface, she said, well, the CD9 tends to be more on the budding kind, for what that's worth. Education Day also reviewed the isolation, and here are 7 methods of isolation. Again, they vary in their quality in terms of recovery, yield, and specificity. So just briefly outlining, we have precipitation by various methods. In this, they're showing PEG, phosphoethylene glycol, I think. And then ultracentrifugation, always been popular in the literature. I think it's not as good to spin your very delicate stuff at 100,000 Gs. You can combine ultracentrifugation with filtration, with density gradients. You can combine a lot of these things. The ones that we use, we believe, are filtered by TFF, or tangential flow filtration, which is a size exclusion thing. Anything too big gets filtered out. There's also these things called density gradients, where you have it go into this solution, and there are different cushions of layers based on their density. Size exclusion chromatography, SEC or HPLC, is a technique used for small molecular detection. It can also be used because of the size difference. The rate at which they come out of the column, it determines their size. So that's often combined with filtration. We have immunoprecipitation or affinity precipitation. We have also things that can dry out and desiccate. And then a newer thing is called AF4, or asymmetric flow field fractionation, which I think uses some form of electricity. So this slide reminds us that they struggled even to know what's going on inside the cell. It's hard. So they do these studies where they add or knock out certain genes, and they see exactly what pathways exist inside the cell. Very, very hard to do, but they're trying. This was what I was alluding to in the purity assessment. When you look at these histograms, the red columns above my head, you see UC is a real underperformer. UC is ultracentrifugation. And if you go to the top second one, you see UC, it's a very wide distribution of histograms. So we have to hypothesize that the ultracentrifugation, the high G, is changing the distribution of your exosomes and forcing them into micelles or binding them, bonding them. It's a mess. So stay away from UC, I guess. ExoQuick is a polymer affinity precipitation. That's number C, nice, tight, high yield. D is TEI, which is a water binder. Again, desiccate and exosomes come out of solution. QEV is an industry standard for size exclusion, HPLC. Ultrafiltration, what we use, also very high yield of exosomes. ExoEasy is a membrane binding system, commercially available. The take-home point is that stay away from ultracentrifugation. Everything else works pretty well. Another thing people were excited about is developing their own labs on a chip, or LOC. What you can see on this chart is they can base it on electrical detection, optical detection. There's a bunch of interesting companies using spectroscopy. This gets pretty sciencey and above my pay grade, but there's something they can do called reading the surface plasmon resonance. And related to that is the surface enhanced Raman spectroscopy. So depending on how the light reacts to what's bound to the surface, it can create different readouts. And in fact, they're creating labs on a chip for single exosomes, which we'll see is very interesting. And so their ability to work at the nanoscale is not limited by the ABI limit. The ABI limit is that light at below 200 nanometers, which are exosomes, you can't see. So they found many ways around this to work with single scale exosomes, even look at single molecules, which is interesting. This is one of the dozens of presentations I went to, and they just, well, they made the point that natural extracellular particles are better because they don't have these problems with penetration of the blood-brain barrier. They are developed or delivered very well into cells, and that the liquid nanoparticles of which the Pfizer and Moderna vaccines were composed, they have some known reactivity of that vehicle. So the vesicle itself is a drug with some untoward effects. They listed out all these different trials of exosomes, some of them engineered, some of them from nature. And then the slide above just shows that you can actually take an endogenous exosome and you can create increased permeability and force stuff into the new package. And then there's other ways you can also hybridize them, etc. So they're pretty clever. They're working on different ways to do this. In the 5 days, there were quite a few clinicians, and this is one example of something brought to the clinic. Uta Erdberger of University of Virginia described the ExoDx prostate IntelliScore. So that's a nice one. They allege that if your PSA is kind of borderline less than 10, but you have an EPI score, which is exosome-based testing, then you really don't need a biopsy because you're low risk. And everyone wants to be helpful and create one of those liquid biopsies. Rudiger Gross of Ulm said a very interesting study, said that why is it that people don't get dengue or Zika from sexual transmission, only from arthropods or mosquitoes? And the answer is because semen and saliva have 10 times the viral particles and actually block and bind the pathogens. Yvette Wolf of Australia National University and Suyong Lee of USC Keck talked about exosomes being created by the retinal pigment epithelial cells and needed to maintain eye homeostasis, the lack of them being very implicated in age-related macular degeneration. Another interesting talk was from Anastasia Tolzunskaya from Moscow, and she said that MSC exosomes from regular human cells can suppress their model of lung fibrosis, giving them bleomycin. Ximing Chang from the University of West Australia talked about the fact that they could simulate tendon overload with their organoids, and they found that when you just stress a tendon too much, it kind of extrudes mitochondria, and that creates macrophages, which bring in inflammation. It's a model for tendon damage and very relevant. And then Leanne Clegg Alborg from Denmark had another one of these lab on a chip fantasies or projects. I mean, she was very stealth mode about it, so I assume they're commercializing it. And her hope is that in the ambulance, as a person is presenting with neurological symptoms, they could triage people to a hospital where they can do ischemic stroke treatment with clot busters versus hemorrhagic strokes. Again, a in-ambulance exosome test for that is what she is thinking of creating. When we look at, again, the holy grail of a lot of what these researchers are looking for is a liquid biopsy. Above me, we have one of the Chinese models alleging that if you look for CA125, HE, or C5a, and you combine them, that it's better for ovarian cancer detection than the CA125 screen. Specificity, of course, for CA125 is 98%. That's high, but the sensitivity, specificity, and detection is very good for their model of exosome-based cancer screening. On the left, we see a bunch of implicated biomarkers. That's usually connotes a protein and or mRNA, and there are some candidates that they can use to try and help with the so-called liquid biopsy. One of the friends I made has a similar conference going on. I guess I missed it, but one of the modules was machine learning. So if you go through and dissolve the exosomes and you analyze them, well, let's say you have 4,000 proteins, 270 sRNAs or small RNAs, and then the machine can find patterns with data mining that you can't, and you can create models. In this case, they're trying to model predictors of brain cancer or glioblastoma multiforme. One kind of cool study from about 4 years ago was published in Cell, and it talked about suppression of exosomal PDL1. Now, we know PDL1 is the programmed death ligand 1 that a tumor cell will put out there to block the T cell from coming and killing it as it naturally does. And they saw something like a 20% improvement in cancer death when they had a blocker. Well, we know that Keytruda, that saved Jimmy Carter or spared him for many years from metastatic melanoma, is often used off-label to kill cancer. And the way it does that is shown to my left. Keytruda or pembrolizumab is a blocker of the PD-1. And so anytime you block the PD-1 or the PD-L1, you have this interference of this innate resistance to cancer destruction. Again, I think people don't realize that the immune system doesn't just fight bacteria, it fights cancer, and that is an interesting finding. So they can modulate the exosomes producing PD-L1 and help to fight cancer. Again, very mind-blowing, above my pay grade, but they have these guys that are basically inventing the technology. On the left, you see like microfluidics. This is actually an integrated circuit they print like you do for a computer chip, and they're able to create these impedance interferometers, and based on the single exosomes, they can be detected, they can be multiplexed onto a readout, they can do fingerprinting, they can create solid gold wells for single exosome testing and molecules on those single exosomes. It's pretty impressive stuff, again, working well below the definition of the visible range. So winding up, one of the last things that we learned about was surface glycosylation. apart from just the proteins in the corona, you can have surface glycosylation, which is a chemical modification adding sugars. And this can, according to this study, really impact the rate and methods of uptake. Important to realize that the people don't really know exactly how they're made, why they're made, when they're made, how it differs from extracellular vesicles, how exosomes are different from ectosomes, if at all. And even once they get in, it's so kind of opaque to us, literally. We can't really know all that much about what pathways they're shunted to, whether they're destroyed. Even the lifetime or life or duration of exosomes is very much in debate. And so anyway, that's me in front of the big sign. It was a really great visit, beautiful city. Was very mind-blowing to see the cool technologies they have. They can paint the surfaces of tissues and cells and exosomes with antibodies, with a DNA paint, and they can see using this fluorescent techniques and others things that are even below the Abbe limit or the ability to discern from light. Most of the companies, well, I would say 5 or 6 were dynamic light scattering. So if you're interested in learning how they count exosomes, go to that one about quantifying exosomes that I created. But the bottom line is when you go and buy a commercially produced exosome product, they're really kind of not sure how many exosomes are in there. A lot of times they're counting particles, which is orders of magnitude more than the exosomes. So it's a case of buyer beware. Anyway, I don't know how much of that you understood. I don't know how much I understood, but a lot of great scientists, brilliant people advancing the field. It's a young field, and we look for many, many great discoveries from them in the future. So thanks, Dr. Ed Park, signing off.